All right, it's my pleasure to introduce Dr. Javier Santander. He's going to be giving an overview of his program, but just as a brief introduction, he's an associate professor in the Department of Ocean Sciences at Memorial University of Newfoundland in Canada. He earned his bachelor's degree in ocean sciences and a master's in microbiology in Chile, followed by a PhD in microbiology from the Center for Infectious Disease and Vaccinology at Arizona State University in 2012. Dr. Santander's research focuses on host immunity and microbial pathogenesis, as well as the design, construction, and evaluation of vaccines for agriculturally important marine species. His lab employs both cellular and organismal models to study infectious diseases and host pathogen interactions in aquatic environments. In addition, his research group contributes to studies related to marine biotechnology, including aquafeeds, probiotics, and functional feed ingredients, and biomedical applications. They collaborate with the aquaculture and pharmaceutical industries, international research institutions, and government agencies to support innovation and sustainability in seafood production. You have a lot going on up there, and we're looking forward to hearing more about it.
Right. Thank you very much, Nick, for your introduction. And we'll proceed to introduce the research in my lab that is focused in marine microbial pathogenesis and immunity in marine cold water fishes. So we are located in the province of Newfoundland, Canada, specifically in the beautiful city of St. John's, where the main campus of Memorial University is situated. Now, the Ocean Science Center is a marine research station located 10 kilometers east from the main campus. This center hosts labs that conduct research in North Atlantic fisheries, oceanography, ecology, behavior, physiology, and aquaculture of marine organisms. So my lab basically focus on the concept of One Health. We study host pathogen interaction in the marine environments under climate change. We study environmental insults over the pathogens or the host in the context of Newfoundland aquaculture. Now, this overview of the Canadian aquaculture. So that is dominated by the production of Atlantic salmon. British Columbia has the lead in the production, following by the Maritimes provinces in the Atlantic. So now here in the North Atlantic, we have emergent aquaculture industry, including landfish, goldfish, and Hollywood. Now in Newfoundland, the blue mussel industry concentrate in the center-north part of the island. In contrast to the Atlantic salmon farming industries concentrate in the south center part of the island. Now, Atlantic salmon production is not exempt of issues here in Newfoundland. For instance, in 2014, we have a severe outbreak of ESIV virus that damaged the production of Atlantic salmon. And since 2017, we are experiencing issues with climate change. So under that context, one of my projects, it is supported by the Atlantic Fishery Funds, that is related to cold temperatures in Atlantic salmon. We're interested what happened during winter in Atlantic salmon. And climate change is not only about heat, it's also about extreme cold. Here in Newfoundland, during winter, we have event of super chilling events, so where the temperature it become below 0 degree during a certain period of time. And that is a huge concern for the farming sector here in Newfoundland. Now, winter mortalities in sea cages are becoming more frequent, and we speculate that the health status of the fish is compromised at low temperatures, particularly below 1 Celsius degree, and might be opportunistic pathogens enhance or cause mortalities. Now, one of the objectives of this particular project was evaluate the microbiome of Atlantic salmon during a winter period. To find out potential pathogens that might affect Atlantic salmon during winter. So this is a summary of this particular objective. We sample in 2 cage sites in the south part of the island during December of cold February. I still remember that sampling, and May during 2024. So this is a summary of what we find. So we sort out first healthy Atlantic salmon versus sick Atlantic salmon that has evident clinical signs. We took tissues of gill, gut, and head kidney, as well sea lice present in the sick fish. Healthy fish have really, really few sea lice during this period. And we conduct a metagenomic analysis of full-length 16S ribosomal sequencing was used in this opportunity. So we find out in all this period, Tennessee baculum was present in the yields of healthy fish, also present in the sick fish. But in addition to Tennessee baculum, the sick fish has primary pathogens like Moretilla viscosa, and certain species of Vibrios were present during this time period. In the coldest months, Tennessee baculum reached the head kidney and might, one of the causative agents of mortality during cold February. So the sea lice also during the cold months has Tennessee baculums in its microbiome. Interestingly, the sea lice microbiome was dominated by Cardiobacteriaceae. We don't know nothing much about this bacterium, but we found this bacterium also present in salmon when sea lice was present. So I'm sorry, in summary, so we can say that during winter, Atlantic salmon is exposed to several cold water pathogens, not only one, several at the same time. So in the context of microbiome, we have another two projects, one supported by the Canadian Center for Fishery Innovation and another by the Atlantic Fishery Funds, focus on wolffish. The idea here is characterizing the microbiota shifts associated to health outcomes, identify potential vertical transmitted pathogens causing larvae mortalities. That is the main issues of wolffish production, which is land-based. Wolffish is produced in Quebec, Newfoundland, and Prince Edward Island, is high-value fish. So this is one example of what we have been recently find out. Wolffish prefer cold waters between 6 to 3 Celsius degree. Warm waters for wolffish are not really good. So we acclimate this fish for long period of time and then study the gut microbiome. And we find out that mycoplasma and malacoplasma, which are beneficial microbes, are dominating the gut microbiome at 3 Celsius degree. As the temperature increase, this population decrease, and we can start to see a domination of Alivio, which is a commensal, and some species are pathogenic. And right now, Ignacio Vasquez is taking over this project and characterizing the microbiome of wolffish at different life stages, including larvae and broodstocks. So this cannot happen if we don't have a really good support. We have the Joe Brown Aquaculture Research building that is dedicated to aquaculture research, marine-focused aquaculture research. This is one example of what they do, is domestication of this particular animal called landfish that is used as a biocontrol for sea lice. This animal actually expose the sea lice on the skin of Atlantic salmon and eat it. So it is some industries are co-cultivated Atlantic salmon with landfish. So to control sea lice infestation, this is one example of the domestication of landfish here at the Ocean Science Center. Now, landfish is not exempt of diseases. It's very susceptible to bacterial diseases. Vibrio anguillarum is the main one. So years ago, in collaboration with the provincial government, we evaluate several commercial vaccines, which none of them works really well. So we end up isolating the strains of Vibrio anguillarum affecting landfish. Here you can see the clinical sign. One of them is exophthalmia in the landfish. So Truncao determined the LD50 in our federal certified facility that allow us to work with even exotic marine pathogens. And so in this recent article, he explained why the commercial vaccine do not work well in landfish and how you can, what you can do to fix that. So taking advantage of that, again, in collaboration with the provincial government and the pharma industry and the aquaculture sector, We evaluate this autogenous vaccine, and the most effective commercial vaccine in landfish. We vaccinate 350,000 landfish and put them in co-cultivation with Atlantic salmon during a year. And evaluate the efficiency of delousing of landfish, and we find out that vaccinated landfish is more effective in delousing than non-vaccinated landfish. This work was conducted by Olo de Junior Onetti, a master's student that currently is a research technician in my lab. So we have here another project in collaboration with NOFIMA and the Research Council of Norway, focusing Francisella noatunensis, that is the causative agent of franciscellosis in Atlantic cod. This is the main pathogen of Atlantic cod. Actually, years ago, shut down the farming industry of Atlantic cod in Norway. So our objectives here are develop an injectable vaccine using proteins of subunit vaccine or a live attenuated vaccine. Turka Paramatan and Raya Gurun are taking over this particular project. So also we have we are participating of this massive project called Marine Biomass Innovation Project, this multi-institutional, very international project in collaboration with the Mi'kmaq indigenous community. So our objective in my lab is basically use marine biomass waste. We're looking for functional feed ingredient to be used in aqua feeds for the aquaculture sector. So Dr. Machin Birige and Douglas Satis are participating of this project. So the most recent project in my lab is in collaboration with ExxonMobil Canada. So we are taking advantage of the deep sea chambers. So we have these instruments in our facilities that simulate deep sea. So we are interested to evaluate high intensity short-term CO2 exposition in marine aquatic organisms and evaluate the effect. Dr. Jaime Sotoneira and Bimba Machimbirique are taking over this together with Luis Tejerina Pardo, who is a master's student that just joined my lab. So the most older active grant in my lab is an insect discovery, focusing in host pathogen interaction and basically Neuromonas salmonicida. Ignacio Vasquez will present more results about that. And just to showcase Aksa Maksud, a master student in my lab, she's studying the effect of melanin. So Neuromonas salmonicida produce this black pigment, melanin. We don't know what is the role in pathogenesis. We purify that pigment, and she's evaluating the effect of the immune response of Atlantic salmon using transcriptomics. And finally, that is the collaboration that I have with Bob Gendron from the Faculty of Medicine. So we are co-supervising Rebecca under the NSERC lead by Bob Gendron. So they are interested in characterize the immune response of teleos eyes to environmental stress, insult, and diseases. They have been found very interesting thing in landfish eyes when it's infected to myxozoan parasite. They haven't found several biomarkers. As well, when the whole fish is exposed to higher temperatures, they have found out effect on cartilaginous growth in the eyes as well. And Rebecca might take it forward after this. So with that, I would like to acknowledge past member of my lab and the current member of my labs that are making everything happen, the collaborators, mainly Robert Gendron from medicine, Dr. Nicole O'Brien, the attending veterinarian from the provincial government of Newfoundland, Danny Boyce, who is the manager of the J-Bar facility, and the funding that came from multiple agencies and private sector. All right.
All right, thank you. You have a lot going on up there. It's very interesting to hear about it. Look forward to hearing more in the future. We don't have time for questions right now, but folks, if you do have anything for Javier, you can put it into the chat or reach out to him after the seminar. Thank you.